Please unmute your mic. You have to unmute your mic. It is muted.
Now it worked. It was not working. Can you hear me?
Yes, yes. So, can you please start sharing your screen so that there will be no technical problem when we start the talk?
All right, and again. You can see my slide, right?
Yes, it's full screen, right?
Yes. I just wanted to check, is the video working?
Yes, it's working.
Okay, fine. Okay, should I leave my video on or?
Yeah, you can leave it on. We will start in 2 minutes.
Okay. I mean, it's okay if I switch it off, right, or not?
No, you can leave your video on. You can mute your mic. When you start, you can unmute it. Otherwise, there will be a echo problem. Okay, am I audible?
Yes, you.
Okay, so welcome you all to the last part of our today's session. And our speaker for the session is Dr. Budhaditya Mukherjee. It is a great privilege for us to welcome a very young and promising Dr. Mukherjee today in this two-day long webinar. He is an assistant professor in the School of Medical Science and Technology, IIT Kharagpur. He completed his doctoral research from Indian Institute of Chemical Biology, Kolkata, and during doctoral work, he was interested in host-parasite interaction and mechanism of drug action in context of antimony-resistant Leishmania donovani infection. He is the recipient of the prestigious EMBO postdoctoral fellowship and joined the Department of Microbiology and Molecular Medicine in the University of Edinburgh. Dr. Mukherjee's postdoctoral work was on host-parasite interaction in the role of post-translational modifications in Apicomplexan infection. He has received the INSA Medal for Young Scientist. He is also the recipient of Pfizer Research Award in the area of infectious diseases, rheumatology, and immunology. So with great excitement, we welcome Dr. Mukherjee to deliver his talk entitled dissection of the fundamental roles played by Apicomplexan Aspartyl Proteases in establishment of parasitism. So, before starting the talk, I just want to inform that there will be alarm that will be going on just before 3 minutes before the end of the talk, that is at 17 minutes. And after 20 minutes of talk, there will be 10 minutes of question, answer, and discussion session.
Okay.
Okay, so I would request Dr. Mukherjee to start his talk.
Thank you so much, Kaushiki, for such a nice introduction. Okay. So I think it's visible. So hello everyone. So as I have been already introduced, I am Budhaditya Mukherjee. I am currently working as assistant professor in IIT Kharagpur. And today I'm going to talk about the fundamental roles played by Apicomplexan Aspartyl Protease in establishment of parasitic infection. So before I go into the topic, I thought that I will briefly talk about my research career, but it's already been well introduced by Kaushiki. So just I will like to point out that during my PhD, I have been always an infection biologist throughout my career. And during my PhD, I work on a kinetoplastida parasite, Leishmania, responsible for causing visceral leishmaniasis on the host parasite interaction part. And after I left my PhD and go to my postdoctoral study, I continued, but in a different infectious disease model, epicomplexan parasites. So what are epicomplexan parasites? So probably you were well aware that the epicomplexa consists of the largest group of intracellular protozoan pathogen, which cause maximum number of death each year in the world, second after the bacterial infection. And it consists of many pathogens which are not only harmful to human but also economically important animals. But I have represented only three of them here. Plasmodium, responsible for causing malaria and 500,000 deaths each year. Toxoplasma, a parasite which is capable of infecting all nucleated cells and affecting one third of the human population. And Cryptosporidium, which in recent years has come up as one of the major cause of child death in India after the rotavirus infection. So I started working on these parasites during my postdoctoral studies. And what I worked on was the infective stages of this parasite. Now, if you look at the invasive stages of this epicomplexan parasite, be it a Plasmodium merozoite or a Toxoplasma tachyzoite or a Cryptosporidium sporozoite, we will see that certain secretory organelle like micronym and rhabdoid are present in all of them. So, like in case of this parasite, as in case of any parasite belonging to the Epicomplexa group, the process of invasion is a highly regulated process which requires synchronized secretion from this secretory organelle, micronym and rhabdoid, which contains several proteins like perforins, adhesins, proteases that allow this parasite to invade their host successfully and thus continue the life cycles. So if you look at in a pictorial way, what happens is this micronym proteins or mixed proteins are released first. Now, as their intracellular parasite, they reside within the host parasitophorus vacuole, and this micronym protein allow these parasites to lyse their parasitophorus vacuole membrane and host membrane, thus allow them to get out of the host, a process that we commonly refer to as egress. And now this parasite started to reinvade a new host, and during this process, they attach themselves with the host surface again, and then they discharge their rhabdoid content. So what actually happens is rhabdoid is again divided into two parts, like non-membranous, but it can be separated as rhabdoid bulk protein and rhabdoid neck protein. So rhabdoid neck protein are released first. This allows this parasite to form a tight junction between its host and the parasite, and then the rhabdoid bulk protein are released or injected into the host, which then goes into the host nucleus or host cytoplasm to modify the host cell microenvironment, thus allowing this parasite to survive. If you look at this process with the help of the video, you will see that these parasites glide, and after they come in contact with the host, they started to form this moving junction. As you can see here, as this parasite is entering into the host, it forms this moving junction and slowly completes the process of the invasion. Now, if you tag a rhabdoid neck protein, where this is a rhabdoid on2 protein, which is tagged with the Encheri construct, you will see something like this. This rhabdoid neck protein moving into the moving junction during the process of the infection, which clearly suggests this important role of this protein in the process of this invasion or the vital part of the parasite life cycle. Now, there is another thing to important to remember that all this micronym and rupture protein, like any other normal protein, they are synthesized in the secretory pathway from endoplasmic reticulum to Golgi, and finally they reach their final destination in the micronym and rupture. So they travel through this secretory pathway. And what happens is in this secretory pathway, this micronym or rupture protein get proteolytically processed, and this processing is not only important for proper functioning of this protein, but also proper targeting of this protein into their respective organelle. So the project started when we were able to discover an aspartate protease, which is specifically present in this secretory compartment where this processing of micronym and rupture protein take place. As you can see here, this aspartate protease is ASP3, which is marked here in red or TITAC, and you can see that it's localized just above the Golgi, which is showing here with the grasp marker in green. So, what are aspartate proteases? So, aspartate proteases are enzymes that are normally present in all eukaryotes. They usually have a pro region and an active site, and within these active sites, there are DTG or DSG motif, which is responsible for the activity of the enzyme. And within this motif, there is flap and flexible loop, which is responsible for ligand specificity. They perform a broad range of function, which include protein degradation, enzyme maturation, signal transduction, and so on. And they have been always a preferred choice of chemotherapy, particularly after the success story of HIV-1 aspartic protease inhibitor, probably the most successful drug for HIV till date. Now, if you look at the aspartic proteases in the broad repertoire of the aspartic proteases in the epicomplexan parasite, you will see that these aspartate proteases can subgroup or divided into several subgroups depending on their function. For example, food vacuole degrading plasmapsins like plasmapsin 1, 2 are clustered in TG. Similarly, TG-ASP3, which function was unknown at that point, was clustered in one group, which is localized in the secretory pathway, and it has two orthologs in Plasmodium parasite, which is referred to as plasmapsin 9 and plasmapsin 10. To study the function of this gene, we approached a classical reverse genetic approach, like to generate a knockdown of these proteins. However, repeated attempt from our side to generate the knockdown of this protein was not successful. You can study this in a simple way. So, as I have already referred to you, that these parasites, once they enter into the host, they grow and replicate within the host for a certain period of time. And when it is time, when the parasite know that now it is time to reinvade a new host, they lyse the parasitophorous vacuole and the host cell membrane. And as a result of lysis, if you stain this host cell with a crystal violet, you see these white plaques appearing. Now, if we see that in case of the wild type parasite, you will see that these plaques are formed normally. However, if you knock out this ASP3, you don't see that these plaques are forming, which suggests that this is definitely important for the survival of this parasite. This opted us to opt for an inducible knockdown system to study the function of this aspartate protease. So what we did is, with the help of CRISPR-Cas9 and a guide RNA, we specifically replace the inducible promoter of ASP3 with a Tate inducible promoter. Now, what happens is, normally in case of the wild type scenario, this promoter is active and the ASP3 will be transcribed and subsequently translated to produce the protein. However, you add anhydrous tetracycline and it will result in a conformational change. As a result, the mRNA will not be synthesized anymore. As you can see here, in case of the wild type parasite, meaning when there is no ATC, you can see that the pro and the mature form of ASP3 is forming successfully, the pro form at the 70 and the mature form at the 55 kilodalton. However, as soon as you add ATC, you started this seeing the disappearance of the protein. As you can see here from the immunoplot, the protein is completely gone. Similarly, from the immunofluorescence, you can see that this red marker, which represent the ASP3 in minus ATC or the wild type scenario, is quite prominent. However, you treat with ATC, the protein is gone. So the first thing that we did after the generation of this knockdown, what is the effect of this knockdown on the processing of the micronym and the rupture protein? So what we observed that in case of wild type parasite, the micronym protein, which is represented here as MIG6, is processed from its probe to mature form, as you can see here in the wild type scenario. However, as soon as you do the knockdown, you started seeing an accumulation of unprocessed micronym protein. Same is the case of a ROP3 marker, ROP1. You can see in case of the wild type protein, it is processed from its probe to mature form. Again, if you knock down the protease, you started seeing an accumulation of unprocessed ROP3 protein. And MIG6 and ROP1 are just two representative of micronym and ROP3 protein. This is true for large number of micronym and ROP3 protein that we checked. So the first thing we look into is what is the result of this accumulation of unprocessed ROP3 protein on the lytic cycle of this parasite. Now, this was studied with a simple assay, as we like to call it an evacual assay. Now, as I have mentioned to you before, that these parasites, as they enter the host, they attach themselves on the surface of the host, and then they discharge their ROP3 content within the host. Now, if you treat this parasite with cytochalasin D, which is an actin myosin motor inhibitor, this parasite will still be attached themselves on the surface of the host and discharge their ROP3 content within the host, but they will fail to move inside the host. So what will you see is something like this. In case of the wild type parasite, they can discharge their ROP3, and you can see this perfect trail of the green trail of the ROP1 marker. However, if you knock down the protease, you see that the ROP3 protein is localized mostly within the ROP3, and these parasites are not discharging their ROP3 content anymore. So we can sum up that this ROP3 discharge results in a defective invasion. So hence, this aspartate protease is responsible for invasion of these parasites. Now, what is the next we studied? What is the result of the accumulation of unprocessed micronym protein? Remember that these two are the ROP3 and the micronym protein that we showed as an example. I will explain this with the help of a simple video microscopy. Now, if we look into the Plasmodium parasite, they can lyse their RBC rapidly as they egress out of their host, right? So they are rupturing the RBC membrane from the intracellular compartment and they're going out of the host. Now, what happens if you generate a knockdown of this protease in case of the Toxoplasma parasite? You will see that these parasites are trapped in a joint ball or vacuole-like structure. They try to break the vacuole and eventually one or two of them succeed, but most of them are trapped in this vacuole-like structure which they fail to break. If I clearly show it here, that you can see here that these parasites are trapped in a joint ball or vacuole-like structure. They fail to break this vacuole and egress out of the host. So in conclusion, what we can say that this accumulation of the micronym protein results in a defect in egress of these parasites. So if we sum up, I have not shown you the data on the plasmid or the plasmodium, but if we sum up, basically what happens is in case of the Toxoplasma parasite, ASP3 helps in the processing of the ROP3 protein, which help in the process of the invasion. And similarly, it also helps in the process of the micronym and the ROP3 protein that helps in the process of egress. Now, this work of ASP3 is completed by two of its ortholog in Plasmodium, where plasmapsin 9 help in the process of invasion and act as a maturase of the ROP3 protein, and plasmapsin 10 help in the process of the egress by acting as a maturase for the micronym protein. Now, as we were studying this project, I have already mentioned to you that aspartyl protease is a favorite choice for chemotherapy, particularly after the success of HIV-1 aspartic protease inhibitor. So, food vacuole plasmapsins like plasmapsin 1, 2, 4, HAP, which have been already well characterized well before, have been always tested as a potential chemotherapy for Plasmodium parasite. Now, as we are working on this aspartyl proteases, a Swiss company, Actelon, which was at that point independent, now a component of the Pfizer, were working on peptidomimetic inhibitor that it have specifically designed against a food vacuole plasmapsin, plasmapsin 2. So plasmapsin 2, what happens is normally expressed 24 hours post-infection, and they are testing the peptide, they are testing the library against the plasmapsin 2. And now IC50, they checked IC50 for two compound, 49B and 49C, of fairly similar structure, you can see. what happened was that these two compounds, both of them have a very low IC50 at 24 hour, that is greater than 500 nanomolar, particular point at which the plasmapheresis 2 is supposed to express. So the company is got not interested in them anymore and dropped them from their screening list. But when we look into their data, we find something very interesting. At 72-hour post-infection, one of the compound 49C has a very low IC50 of zero point 0.6 nanomolar. Now, to mention to you that this zero point 0.6 nanomolar IC50 is lower than artemisinin, which is currently used for the treatment of the plasmodium. And this 72 hour is roughly the time where this late stage plasmapheresis 9 and 10 are expressed. So we got interested into this compound 49C and decided to study it further. Now, if you look at the aspartate proteases that are expressed across the erythrocytic blood stage of plasmodium, you will see that the food vacuole plasmapheresis 1, 2, 4, and half are expressed from the early ring to the trough stage, which is the feeding stage of the parasite. Plasmapheresis 5, which is mainly responsible for cleaving the protein, which are then transported into the host cells, are also roughly expressed during this time. And the late stage plasmapheresis 9 and 10, are expressed during the schizon stage. So to characterize the compound, what we did, we applied the compound either on the early ring stage or on the early schizon stage, and then allow this parasite to go through their life cycle, to complete their life cycle and recollect the parasite when they break this mature vacuole and reinvade a fresh RBC, which is characterized by the formation of this immature ring. So what we observed that when you apply the compound either on the early ring stage or on the early schizon stage, the parasite get blocked in the schizon stage, meaning they are not able to break this vacuole and reinvade a new host. Whereas the DMSO treated parasite, if you can see here, that they can successfully reinvade a new host, which is characterized by the formation of this immature ring, whereas the 49C counterpart cannot do this. Similarly, if you break this vacuole mechanically or rupture them mechanically with the help of a syringe and then allow this parasite to reinvade a host, they still fail to do so. This clearly suggests that this compound is targeting both the egress and the invasion of this parasite. In turn, it is targeting both the plasmapheresis 9 and 10. It is also important to notice here that the parasite, when the compound is applied on the ring stages of this parasite, I mean in this stage, it does not stop the parasite from developing into a mature schizon. So it clearly suggests that this compound have no effect or no visible effect on the other plasmapheresis that is expressed in the other stages of the erythrocytic stages of the plasmodium infection. So to convince you visually that this actually happens, let's look at this video image where basically what we did is we tagged a PV protein, which is PV1, with the GFP. Now, this results in the accumulation, this protein results in the accumulation in the parasitophorus vacuole. So these are intracellular parasites. So you will probably aware that they grow inside the parasitophorus vacuole of the cell, of the host cell. So this protein accumulation results in the accumulation of the green signal within the host. Now, as the parasite lies the host, in case of the DMSO treated parasite, you can see that you were losing the green signal spontaneously. Now, what happens when you apply a low concentration of 49C, which in this case is 1 nanomolar? You see that this green signal is accumulated within the PV, and at some point this is gone, but the parasite is still maintaining within its circular structure. This clearly suggests that at low concentration, the parasite is able to break the parasitophorus vacuole membrane, but it fails to rupture the host cell membrane. So the parasites are still trapped into the host cell membrane. Now, if you applied a higher concentration of the drug, you will see that in that concentration, the parasite remains trapped within the parasitophorus vacuole and it is not able to escape within the parasitophorus. It is not able to escape even the parasitophorus vacuole membrane. So the green signal is persisting throughout. Finally, we proved this also in the animal, in the P. Bargai model. As you can see here, normally if you infect the parasite, here it is infected with the sporozoite or Plasmodium burgei, which is the mouse strain of this parasite. You see that this is the luciferase expressing strain. So at the 44-hour post-infection, you started seeing the signal accumulating in the liver. So this is blue basically represent the hepatic cells which are infected with the luciferase expressing parasite. 52 hour you get a kind of similar picture Now at 55 hours, the signal disappears. This is probably the time when this parasite started to leave the liver cell and start the blood stage infection. At 59 hours, you see that the blood stage infection is starting to appear slowly, and finally it spread throughout the mice, as you can see here. Now, what happens in case of 49C? So 49C was injected into this mouse 20 hours post-infection, and you can see that similarly to the white type control, at 44 hour you see this blue signal appearing in the liver. At 52 hour, there is a more instant signal in the liver. This is probably because this parasite is failing to egress out of the liver cell, which is evident from here. At 55 hour, when the parasites escape the liver cell and started their blood stage infection, in the instance of 49C, they are failing to do so. So you still see the signal in the liver. Finally, the signal in the liver started to disappear. However, this parasite failed to invade the blood cells, and hence the mice is cleared of the infection. So, in conclusion, we can say that we have two compounds, 49C and 49B, of fairly similar structure with certain difference in the aromatic ring, and one of them specifically is target this plasmapherin 9, thus helping in blocking egress and invasion of this parasite, where with a slight different structure, it fails to do so in case of 49B target, which again point out to the high specificity of the compound towards this plasmapherin. Now, with this, I would like to acknowledge my PhD lab. I haven't talked about any of my PhD data in this presentation, and this is all of it was done in my postdoc lab, and Professor Dominic Soldati's lab in University of Geneva. And this is the old building and this is the new building, and as you can see here, was my office for more than 4 years. So it was really nice to talk about this work, and thank you all of you for listening, and stay safe. With this, I will conclude, and you are free to ask any question.
Thank you, Dr. Mukherjee. That was a really interesting talk. So now we would move on to the question answer session. So I would request Junaid to moderate the question answer session. Junaid is here. Junaid, hello.
I think he's muted. I cannot hear.
Junaid, please unmute your mic.
Hi, I think on behalf of Junaid, we can start the question answer session. Yeah, if any DLS faculty that they want to ask any question? Okay, then we can take some question from audience. Abhishek Bandhan, he is asking, aspartyl protease expression regulated during invasion and aggression of the apicomplex proteases into and from the host cell respectively.
I think what I understand is you were suggesting that whether these aspartyl proteases have a particular time profile of expression, meaning whether they were expressed more during the time of infection or not. So to answer your question, so you see that this when I talk about the plasmodium, so you see that the plasmapherin 1, 2, 4 were expressed at a particular time point, since and the plasmapherin 9, 10 are expressed during a late time point. So yes, these enzymes are expressed only when they are needed. So they have a specific time regulation of expression. Now, the most of the substrate of these proteins or these proteases are rhabdoid protein and microneme protein. And rhabdoid protein and microneme protein need to be functional at certain point. However, they are produced in a manner that they can get processed and reach their final destination. So to answer to your specific question is that aspartate proteases, yes, they are regulated in a timely manner. However, there if there is a constitutive function, then it is produced throughout the cell, throughout the parasite life cycle in a controlled manner.
Okay, thank you. And he has another question, that is the region encoding the aspartyl protease conserved among the epicomplexes which infect the plant host as well?
Well, it depends on the function of the particular aspartate protease. They are conserved. For instance, in Toxoplasma, Plasmodium, Cryptosporidium, these plasmapsin 9, 10, and you will see that Astri and Cryptoorthologs all have a conserved domain, particularly in their catalytic site. In the region of their ligand binding specificity. And yes, they are largely conserved and they are largely different from the host or the eukaryotic aspartate proteases like we have in our body, like renin. So that is why they are always the preferred choice for chemotherapeutic targets.
Okay. Next question is from Orpita Datto. She's asking, is there any prototype designed the proteases that can structurally and functionally inhibit the protein from acting? However, is this mechanism of development reliable with other similar ones?
Okay, so I have not presented that one, work. So that was also we did. That was one of my work during my postdoctoral research. So we tried to explain the structural functional basis of aspartate protease. So specifically, what we try to answer is why a particular compound 49C can target specifically plasmapsin 9, 10, or ASP3, and it failed to target a closely related aspartate proteases, like is plasmapsin 1, 2, or other food vacuole plasmapsins, although they have quite a region of conservation in among these aspartate proteases. So what we come out that there is a conserved phenylalanine residue in the aspartate proteases, which if you mutate to a tyrosine residue, that can make this aspartate proteases susceptible, or then can make this aspartate proteases rather resistant to this particular 49C. So actually there is a structural and molecular basis of answering this question, but I haven't talked about that data in this presentation. So to simply answer your question, yes, you can design drug specifically targeting this particular region of aspartate protease, which can be utilized for preventing the infection.
Okay, thank you, Dr. Budhaditya Mukherjee. I think this is from the audience, and if any DLS faculty want to ask any question, they can go ahead. I don't think anyone would like to ask anything. Behalf of the organizing committee, I would like to thank Dr. Budhaditya Mukherjee for the fantastic talk, and to the audience that tomorrow we will be live from morning 9 a.m. So stay tuned with us and stay safe. And this is, we are signing out from today. And thank you everyone
Thank you
for
so
joining
much.
with us. Thank you everyone.